Please join me in welcoming to the Distinctive Voices podium, Dr. John Rinn.
All right. Well, thank you, Monica, and thank you. I can't believe there's this many people that want to hear about junk DNA. I am flattered, honored, and it's a real pleasure. So I don't know if you can. The slides are coming up. There we go. What I want to talk today about is what our research is doing to better understand this. The human genome. So how many people in here, I know it said that you guys can't ask questions during the talk, but it didn't say I can't ask questions to you. And I teach undergrad, so I can wait a long time. So how many people know this term, the human genome? I've heard it. Okay, good. That's what I figured. It's this elusive term that's used around, they've sequenced the human genome. And when I was a graduate student in 2000, the human genome was about to be sequenced. So we were going to have all the sequences that make up our DNA fabric that can produce all the different cell types in our body. And it was almost as if this pie was going to fall out of the sky and tell us all the answers about humanity and why we're special. But the answer was kind of a bummer. It turned out we were only using about 2% of our DNA. And this reminds me of the old saying that we're only using 10% of our brain. And I hope somebody starts studying that, because there's got to be more to that than that. But we're basically we're using 2% of our genome, and the reason we thought that was that conventional wisdom was that DNA is the template. It's present in all of our cells, makes up the instruction sets for all of our cells. But it makes that by producing a transcript of RNA, which is just a mere messenger to be sent off to be turned into a protein, and that the proteins go around doing the main jobs of the cell. But if you look at it just a little bit different, this is my movie. If you look at it just a little different, you get this. You get the rest of the pie. Although only that 2% of the genome can make these precious protein coding genes that are really powerful genes that we've been studying for many years, when you look at it from an RNA view, the whole genome sort of opens up. It has the potential to make RNA, it just doesn't have the potential to make proteins. And what we're going to learn, hopefully by the end of the lecture tonight, is that RNA can actually come back and influence the DNA and tell it what to do. It can go find proteins and bind to them and actually tell them what to do, and it's not just a subservient messenger. And in fact, once it binds to those proteins, it can even tell them how to get back and work on DNA itself. So this one simple linear flow of information that we thought made a lot of sense has now become more of a circular flow of information. So we'll take a step back and test you on a little bit harder question. Which of these do you think is an RNA gene? How many people think it's A? Good. Okay, I will note one person raised their hand. How many people think it's B? Okay, this is interesting. Okay, how many people think it's C? Okay, good, yeah. So, wow, you guys are very, I'm impressed. Do you need me to tell you guys anything else? It sounds like you guys might already know all this. But it is true. So C is a non-coding RNA. It's one of these small RNAs called a microRNA, and it actually serves to regulate the messenger RNA that's going to build a protein. But congratulations, I think you're the only person that raised your hand for A, is an RNA gene. However, I can see why that was tricky, and some people did raise their hand for B because it looks just like an mRNA. So this B is going to make a protein, and A is not. But you really can't tell the difference just by looking at the two. And so what we call these are long non-coding RNAs. And like mRNAs, they're split and stitched together to make a final transcript product, but they don't go to encode proteins. And I also should know that you guys probably already know that there's lots of RNA genes in the cell. So I don't want to make you think that these are the only thing going on here. There's many of them that we've classically known for many years and play very important roles. But more recently, we've found that there's lots and thousands of these other types that look just like an mRNA. And that's what's been sort of intriguing or mysterious about them, is why do they look so much like something that should be making a protein but acts as an RNA? And so that's what we're going to talk about and focus on for tonight. I might call these things accidentally link RNAs. That stands for long non-coding RNA, sort of a shorthand for it. And they come in different positions in the genome. So sometimes these long non-coding RNAs are actually encoded inside of a protein coding gene here and goes in the other direction, called an antisense RNA. Sometimes they come from within the introns. We once thought, once splicing was discovered, introns were just discarded and became garbage. Actually, some of these introns encode RNA genes as well. Some of them are bidirectional, meaning that the protein coding gene, it shares a regulatory region with it and is sort of transcribed in the opposing direction. And intergenic, which is just another variant of this bidirectional one, where the RNA gene sort of sits in the middle of nowhere. I grew up in Minnesota, and this is a terrible analogy, but people always say, never shoot a deer behind a tree, wait till it's out in the field. And so we've been targeting these guys because they're sort of sitting out in the middle of a field. If we go and manipulate these genes, we worry that sometimes the effects we will see will be due to messing with some of the protein coding gene as well. So this is just a very preliminary first place to start. And go from there. But really everything I'm going to tell you applies to all these types of long non-coding RNAs. So I think we're ready. I've had enough background to enter the wardrobe into a place I fondly call non-codarnia. It was one of my favorite childhood books, of course. And this place, so we're going to go through the wardrobe and explore what we've sort of learned in the past few years about this space. And so this is what it looked like to me as a naive young graduate student in 2000. My advisor said, well, there's all this junk DNA. Go see if there's anything out there. So this is kind of what it felt like. It was junk DNA. Nobody really cared about it, but it was kind of nice, quiet, spacious, sort of like the running trails around here, open area. And the human genome sequence allowed us to then go and look for biological activity. So essentially RNA is the first step in understanding or making something biologically active. And so we reasoned that if we can go find RNA in these junk regions, that maybe we can go see if they're doing something. So we did that, and quickly, us and many other groups quickly found that there's the non-coding genome is riddled with RNA. So it quickly went from this nice, vast, simple place to something that looks more like this, from Jabba the Hutt's palace. So we wanted to go in and understand these mysterious creatures, and whenever you're in a mysterious, bizarre world, the thing you really want to have is a map to help guide you around and figure out where you are. And this has been a hobby of mine. It's almost like stamp collecting. I always wanted to name a star as a child, and now I get to name genes, so it's fun. So we can take this map of all this different RNA, and we've been doing this for over a decade with many very impressive students shown here. And what we're looking for is our target is a gene. Okay, so these things look just like the messenger RNA, so let's go find things that look just like messenger RNAs that don't make proteins. And this is a way of packaging DNA that indicates the start of a biologically active segment of DNA. It's called a promoter. And the way it's packaged is indicative of that type of functionality. And then right next to it is packaged a different way with a different histone modification that tells you where the gene starts and where it ends. And so we can do that plus overlap RNA sequencing data to build the transcript structure that resides in this region. So we can essentially, and what we do is we take a map of the genome and just sort of walk along it, and we say, oh, well, these are protein coding genes. We're not interested in those. What about this guy? What is this thing that's sitting out in the middle of nowhere? Nobody's ever annotated it before, and it doesn't seem to make a protein coding gene. So these are what I'll be referring to as link RNAs or long non-coding RNAs, and there's at least 8,000 of them. It depends on who you ask, 10,000. It could be upwards of 20,000. But I think that raises sort of a question that might be in the back of your head. If there's so many of these things, is it noise or is it functional? And this is a really important sort of thing to think about, and it's sort of been a chip on my shoulder for over a decade. And that just because there's biological activity doesn't mean it's doing anything. It could just be that the transcription machinery made a bunch of accidents and now we're seeing them, or it needs to roll through this desert to get to where it needs to go. There's lots of things you can rationalize, and really what this debate boils down to is the fact that we know very few examples right now. I'll show you a couple in a minute that have been known classically, but the ENCODE consortium, or the national consortium to sort of annotate the genome, said basically that 80% of the genome is transcribed into RNA. So there's a real bandwidth gap here between what we know is functional and what we can see. And so this noise versus function debate raged and still is ongoing today. And instead of getting wrapped up in it, we decided to just take a totally different approach. By now there's probably more than 100 known genes, but still far less than the number of transcripts we can see and detect. So instead of getting involved with a numbers game, I realized quickly you never want to put a number on anything because you'll eventually be wrong. But people are still trying to do it, and they're trying to figure out what this is, and we sort of changed our direction and said, let's just find ones that are functional. Who cares how many there are? Let's just find some and start sifting through them. It's almost like sifting for gold through sifting pans. And I also just want to point out one other point, is that there's a difference between junk and garbage. We all probably have this somewhere in our house. I have one of these. And they're actually kind of fun, right? They're like this drawer where you put stuff that you might need one day. You're not totally sure that, that love letter or whatever might come in handy. So you keep it around, but you don't use it very frequently, and it's used once in a blue moon, or you're like, aha, oh, there's that thing in the junk drawer. So maybe the genome does have that, and a lot of the ones we don't see function for could be just storage for an appropriate time that we haven't detected yet. And that's the difference between garbage, which is something smelly and nasty, which you're just trying to get rid of. So we took a different turn and really wanted to understand, are these things functional? Do they do anything? If we manipulate them, will they change the way a cell behaves? And so really what we want to do is we know how cells differentiate from, let's say, a stem cell into a brain cell or a liver cell, and the protein factors that drive and define these cellular transformations. But we don't know how these guys play a role. And so we want to see how they fit in this equation. So why would we even think this is worth doing? Lots of ways this could go, right? But let's take an example. So what's the difference between Bill and Hillary? You don't have to answer this one. Actually, please don't answer that. But they're both, very prominent politicians. They both like taking pictures next to flags, which I'm very, look at this. I definitely will not be running for president. But but Hillary has a trick up her sleeve. She expresses one of these long non-coding RNA genes that I've been talking about that gets spliced together and can't encode a protein, and this single RNA gene can crumple up and shut down an entire chromosome. I mean, that's really a power to behold, that when people say it's all junk or noise, it just can't be. We know at least one of these things exists. Now, maybe somebody's playing a prank on us and said, let's just give them one and make them think the rest are important. But my bet's on there's got to be at least a couple more of these. And so this gene exists that Hillary has or females have to give us guys a fair chance, is because females have 2 X chromosomes and they want to shut one down to make the balance equal to us guys. And the way that's done is it's caked with RNA and it gets excluded off to the outer periphery of the nucleus. That exclusion to the outer periphery of the nucleus will actually come into play to being important later in the talk. And I'm also going to give you a teaser that there are other ones like it. This is one called FIRE, which we just recently published, but I'll talk to you more about at the end of the talk. I just want to give you a teaser. But it's also on the X chromosome, but it makes very large foci on both X chromosomes and not just one. So the way we've been tackling function is to take we want to start with the human first, obviously, and sometimes our best proxy is to take human regions and make mutant animal models. This is like the ultimate test. This is very expensive, very laborious, very, very difficult. But it is kind of the final answer because it's genetic manipulation of an animal and you can see what happens. So in the meantime, we tend to do things in cell assays where we can toggle RNAs up and down and ask if they affect the way a cell is supposed to behave. And then, of course, we want to get down to the molecular level, understand how they work and move and what the moving parts are. And this information flows back and forth, and projects go all sorts of different ways. But what's interesting is in the past 7 years, I would say, or maybe more, at least in the past 7 years for me, you can't do this anymore without a computer. You simply can't survey 3 billion bases by eye. It's just more and more molecular biology is requiring computational biology. And it's a real fun transition to see new fields of people in astrophysics and all sorts of other places where computer programming has played a role moving into biology. It's an exciting time. So here's an example of how we can spin through cell systems and quickly identify some candidates that do have functions. There's great hope now that we can have personalized stem cells, that you can take one of your skin cells and turn it into a stem cell by adding some factors to it. This process is well defined, and we can ask how these guys play a role. I won't give you the details, but we screened through about 40 or 50 of these things and found a couple gems. This is a long non-coding RNA we call ROR, for regulator of reprogramming. I was trying to go with the Lion Witch in the Wardrobe theme, but it quickly failed. But so normally when you reprogram skin cells into stem cells, you get about 1 to 5% of the cells can do this in a dish. But if you don't have this long non-coding RNA, you absolutely can't do it. It abolishes the process, so it's required. But what was surprising to us is that if you add more of the RNA, you get more stem cells. So we can start to think about how we can manipulate these RNA genes to help produce more stem cells, but also get the notion that these things are critically required in these cell-based processes. And there's more like this. This is just one example. Another one that's kind of exciting is adipogenesis. Again, another very well-defined system. Believe it or not, people can make fat cells in dishes all day long. I don't know, that's not something we don't usually try to do in our bodies, but you can use this system to understand, again, gene regulation, where there's well-defined factors that make this transition from a precursor to an adipocyte or fat cell. And this is what it looks like on a dish. You can put a red dye, and it stains wherever there's an adipocyte. And so you see a red dish when you get to the end process here. But as you probably guessed, you take away one of these long non-coding RNAs, you absolutely can't make the adipocytes anymore. And this turns out to be fire, the one I'm sort of teasing you about that we'll get to at the end. We found it through this screen. And using these kind of systems, we can screen like up to 40. Now remember, there's thousands of them, but we still only have handfuls of functional examples. So we're trying to be practical. I call it mesoscale. It's not quite high scale. It's not quite low scale. It's somewhere in the middle, or mesoscale. We can even start to tackle malaria, where this parasite is quite fascinating. It has a life cycle where it develops in our blood and then it gets angry and then goes and invades more blood cells. But what's weird about this parasite, it has very few factors that allow it to transcribe its own genome. And we've been exploring this for the past several years with Pardeep Subedi and a joint student. What we found is that their genome is also producing many long non-coding RNAs that may be even interacting with human host cells. So this is not limited to humans. Almost every species seems to have these long non-coding RNAs and playing very important roles. In fact, we're very interested in one that seems to be turned on exactly at this stage of invasion. And you can imagine maybe making RNA blockers that would be able to stop this process from happening. That wouldn't be toxic to us because we don't have malaria genomes in our body. But really, this is kind of nice, and there's many other cell systems we've gone through, and we did this, and criticism started to come as well. Okay, fine, this is all in a dish, it's in plastic, humans don't live on plastic. What is this really going to tell us about our health and human health and disease? And so, again, based on lots of criticism, we took another sort of turn, and we really wanted to get down to the genetics and to the animal level of how these things contribute to our human health and disease. And so really this advice came from Ed Lewis, my favorite geneticist, where he said, the laws of genetics have never depended upon knowing what genes are chemically and would hold true even if they were made of green cheese. And I think that's really the right way to think about this, because if you take away a piece of DNA and something happens, it's important. How it works is a different question. And that's probably the hardest question to actually figure out, is how is it working? But this really hit me over the head and said, you know what, let's take a step back, forget about the RNA. Let's use the RNA to help us find regions of the genome that are active, that might play a role in human health and disease. And so you can just simply cross these two maps, these maps we've been making of these long non-coding RNAs, with the ever-growing population genetic studies that are going on. You can take cohorts of people who have a disease and people who don't have a disease and find the risk susceptibility loci for that. For instance, how many people have done 23andMe or Me and 23, whatever it is? I did it, if that helps. So a few people have. So the genome is becoming quite personal, and that you can really figure out a lot of stuff about, like for instance, it told me I had brown hair, brown eyes, and it's quite accurate what it can tell you, but what I would have never guessed is that I have a risk for melanoma. I have brown hair and brown eyes, right? So I shouldn't have a problem, but it turns out doing this test told me I have it. So by studying the human population, we can find variants in people and where they're susceptible for certain diseases. What was remarkable is when you do cross these two maps, 400 disease regions that are strongly associated with the disease have no protein coding genes in them. These have typically just been thrown down to the bottom of a graduate student's pile of to-do lists and usually rise right to the coolest looking protein that's nearby. And so what we can do now is these all have long non-coding RNAs in them. We can go target these, and we're down to a reasonable number that I think we can start to approach and see how this will help us in our future. So what we had to do, though, is then take that morass of RNA transcripts, cross it with those genetic regions, and then filter even further by making really sure that they don't encode a protein. I won't go into all the details, but using multiple layers of evidence, we can guarantee they don't, or our best proxy, they don't have a protein in them. We also want to make sure that they have a mouse and a human ortholog so that they're the same gene in human and mouse, because we're going to go now take it out of a mouse and ask what happens. But if it was only in the mouse and not in a human, you probably would fall asleep right about now. And in fact, that happened to us. We had a really cool gene. We found it. We'd studied it in mouse cells. And it turned out there was no human equivalent. And people are just kind of like, yeah. So always have a human ortholog is always a good thing. And so we can, So we did this, and we found this sort of harkens back to this debate of whether this is functional or not. We picked 18. And that sounds small, but it's humongous. It's the biggest risk I've ever taken as a scientist. And this is probably the most rewarding table I've also seen as a scientist, is that many of them, or 3 of them, are required for life. So if you take away a piece of this junk DNA and you die, you really got to start to think about a different definition or think further about what these things might be. Now, we don't necessarily know it's an RNA yet, but the RNA told us where to look, and the human genetics told us where to look, and this combination was quite powerful. We also saw a bunch of other interesting phenotypes, which I think I'll summarize later. But there's all sorts of funny things, from mice that are missing parts of their brain, but happen to be totally fine. They live normal lives, mate, and eat just fine, even though they're missing regions of their brain. We have some that have very weird behavioral problems. Some that are sterile, and many other different types of phenotypes. So these things don't seem to play one simple role. They seem to be involved in a wide range of different processes. And this is probably the most potent one we found, shown here. And this really, I think, highlights the power of using this approach. This is a disease called alveolar capillary dysplasia, misalignment of pulmonary veins. I will not refer to it again because of that long name, but it's been typically thought this disease is caused by this protein coding gene called FOXF1. But more recently, as people have been studying patients' DNA, they found a recent paper published, about 3 or 4 years ago, that published a paper that said, actually, there's patients that don't have any deletions to this protein, but they only have deletions in this non-coding region, which encodes one of these long non-coding RNAs shown here that we call Fenderer. We call it link FOXF1 also. So we knocked this region out in a mouse, and we left in its promoter so it could stay biologically active, so we could rule out just the fact that transcription itself was doing this, and deleted the majority of the thing that would be made into the long non-coding RNA. And in its place, we left a little reporter gene so we can track where it would have been in the body. And so when we do that, we can see it's expressed very highly in the lungs. It's very specific to the lungs, actually. And which is fitting with the disease that this region was found with. And in fact, when you knock it out, not only do the mice die, they die at birth from lack of breathing, and they have many of the same things that are sort of seemingly similar to the human disease. So basically, we can use this approach now to find genes that are so potent they kill mice, and then try and take these sick animals. Well, I really messed up the ordering of that. But basically, I think this shows that they're functional, B, they're important in human health and disease, and we can use animal models to sort of get at the very basic preliminary understanding of how they might be working, and then also try and fix these mice to see how that could translate into human therapies. So this was one example. We found many, like I was mentioning, ones that are even little tiny guys that we called midget, and many other different things. But the fact that you can take away a piece of junk DNA and not be able to live, I think, speaks for itself. So now that we have all these mice that are sick, we need to do something about that. That's not very nice to them. So we were taking some of these mice that are lethal, and we're bringing them to the hospital. And we're doing this in collaboration with Pier Paolo Pandolfi, who's a venerated cancer biologist, and he's really done remarkable things in cancer. And he had the foresight to develop a mouse hospital. I thought that sounded funny at first for a year or two, and then when this happened, I realized, wow, he's the only guy who's got a mouse hospital. You know, who do you go to? And this is at Beth Israel Deaconess Medical Center, and so we've teamed up with Jeff Saffitz and other people to try and utilize this resource he's built to understand how we can help fix these things. And it's amazing when you see this facility he built. They probably get better care than I do at Mass General Hospital, not to mention any names, but the mice are very well treated, and it sounds funny and silly, but it's actually really important, because if we're going to now that we know there's these RNAs or these loci are involved in the disease, we want to know exactly when, where, how, why they come up, and then how we can use other genetic models and other treatments to try and actually fix them. And RNA is not the best drug in the world, but we're trying our best to use it to try and fix these diseases. So in summary for this part, we're using the approach of finding disease loci in the human population where there's long non-coding RNAs, and then we layer on more and more evidence that we've gathered from cells, that we've gathered from other people's data. We like free data. We use public data all the time. And so we've been able to accomplish this, and this is where we want to be, is right here. And we've been able to do this at least in one pass, and we're continuing to use this approach to cycle through more and more and find out what other diseases they might be involved in. So now that it's worth figuring out that they're important, now it's time to figure out how do they work. How are they moving on a molecular level? And so the one I want to tell you about today is this lincRNA fire. But first, I'll give you some background. There's some ingredients. We're going to have a cooking class here. RNA is one of the most devious, recluse molecules there is known. It doesn't follow any rules. It always blows our mind. It does weird things. It's even able to do catalytic reactions itself. But there's only one rule it always pretty much follows, and that it binds to a protein. It really doesn't tend to work on its own without a protein at some point. So we know RNA can bind to proteins. We know RNA can bind to DNA, and it can do that in two ways. It can wrap around the DNA that's in a helix, or it can actually make complementation to the sequence that it came from. We know proteins bind DNA. In fact, this is how most gene regulation occurs, is a protein comes in and tells a gene to turn on. And we know RNA can bind to RNA. This is how splicing works. When you stitch these genes together, it's actually governed by an RNA-based recognition process that then proteins come in and put it all together. And so if we combine these things, we can make some really cool creatures. So if you add A and B, so an RNA binding to a protein and DNA binding to RNA binding to DNA, you get this regulatory mechanism that's been found here, where the RNA is helping tell a protein where to land and when to be on and when to be off. So that RNA sort of serves as a landing pad for a protein to come in and turn a gene on. If you add A plus C, you get what we call the scaffold. And this is really interesting, and many of the long non-coding RNAs seem to function this way. Is that imagine you have a process that requires 5 or 6 proteins. You never seen these this new middle school or kinder school phenomena where you have like a bunch of kids on a string and there's a teacher walking them? I didn't have that when I was a kid, but it looks fun. So that's what this RNA is actually doing here, is it's stringing along a bunch of proteins to get them to the right place and sort of chaperoning them in there. And we call it the scaffold, and it exists. Hillary Clinton's gene does this as well to carry out its function. This is my favorite, second favorite actually. If you add A plus D, you get the ribosome. The machine that makes the proteins is an RNA gene. That just won the Nobel Prize, that the ribosome is a ribozyme, and then it's actually an RNA functioning to make the proteins. It was kind of a remarkable finding. But again, it's just simple ingredients. You add RNAs binding to proteins and RNA binding to RNA, and you get this massive ribosome structure. And this one's the most fascinating, where you add A plus B plus C, you get not only the scaffolding, but you get the ability of the scaffold to have the RNA tell the scaffold exactly where to stick to. And this is telomerase, which makes the ends of your chromosomes stay the right length. Essentially, aging is defined by the shrinking of your telomeres. It's almost like the rings in a tree trunk. You can see age by shrinking of telomeres, and this is the machine that makes sure they stay the right length after every cell division. So it, again, is an RNA machine, and it uses the RNA to know exactly where to go and pinpoint the spot it needs to go to. So again, you can see that this isn't going to follow one simple rule, and you can make all kinds of different devices, if you will, using these principles. And so another way to put it is they're RNA tinker toys. That you can use RNA to think of the wheels as proteins and the rods as RNAs. You can start adding these things up, and you can build up really interesting sort of complexes, and that's what we're starting to see happen quite often in the cell. So now these complexes can do things to DNA, as I mentioned in the beginning, where the RNA serves as sort of a little beacon where it opens up, it makes an RNA, and that RNA can actually tell the proteins to come in and bend DNA to bring another gene close by into proximity. So this gene could be, let's say, a mile away in distance, but the activation of this RNA has caused it to loop and come into proximity because this RNA is going to help this gene turn on. It's called an enhancer. We've known about enhancers for years, but we've never known that the RNA that it needs to be transcribed, and that the RNA itself has actually been shown to be required for this folding, not just the transcription of it. And so this brings us to FIRE, where this can also happen in 3 dimensions. Rather than just looping one chromosome around, we can actually maybe bring multiple chromosomes together. So back to the teaser I said before, this is this powerful gene that shuts off an entire chromosome. It's required for females to live. And we found this other gene that's on the X chromosome, makes one of these big clouds, but does it on both X chromosomes. And that really struck us over the head. Actually, I'll never forget when I was at our departmental retreat 3 years ago when this picture was sent to me, because we sent a buddy of ours who takes pictures of genes, because seeing is believing. A lot of these things you can't see, so we don't want to study the ones we can't actually see in a cell. And so we sent like 40 of them to a friend of ours to take pictures of, and he sent this one back and said, "Whoa, what is this? Are you sure you didn't hit, are you sure this doesn't exist?" And so we got really excited about this one and dug all the way in. And so the way Arjun takes these pictures is he developed a method where you can use a green-colored probe that will stick to the RNA. And that, if you put it in the introns, you'll know where the gene's locus is, because the introns only exist at the time it's being transcribed. So you're essentially pinpointing where the gene is in the genome. And then the red exons tell you where, after it's been put together and it's mature, where does it end up in the cell. And so you can see it starts out here. These are male cells, so they only have one X chromosome, so we only see one dot. Here's the mature transcript. So as you can probably guess, they are overlapping. So this RNA seems to form a cloud around the site where it's transcribed, but it seems to be bigger. It seems to be spreading a little further out. And I'll show you some more pictures of that in a second. And this is in female cells, where you see the same thing. Now, what's interesting about these are stem cells where the X chromosome has not yet been inactivated for females, but this cell it has been. And so here you have still, this gene is being transcribed from both X chromosomes, even though one of them has been shut off and pushed off to the periphery. And if you look at this, okay, so this is the second movie. It's really just two pictures overlaid again. But you can see it sort of go from its site of transcription and sort of spread out from that region. That was really fascinating to us because the only other time we had known about that was through this exist gene. So we wanted to say, where's the fire? Where is this thing actually touching DNA? And so what you can, we collaborated with Mitch Gutman and Eric Lander and Jesse Engritz to use this technique they've developed where you can build oligos that will grab an RNA, shown here. So it'll attach to the RNA, and then you can pull on these handles and find all the DNA that's sitting next to this RNA. So essentially it's a genomic footprint of where this RNA touched DNA. And what was perhaps not surprising is, since we know this is on the X chromosome from the green dot, we saw it spread across 5 million bases of the X chromosome. So it started in one spot and spread for 5 million bases, or 5 megabases across the X chromosome, shown here. This is where it starts. And it sort of did so symmetrically in both directions. So what was really surprising is we found that it touched 5 other chromosomes. If you think about this for a second, there's 2 dots, there's 5 chromosomes. Why don't we see 7 dots in this picture? This was confusing, but I have to be honest, it wasn't immediately obvious. And we just sort of saw this data and we're like, oh, the data looks good, it's legitimate. It was just a moment of like, wait, there's only 2 dots. How can this possibly be? And so we call these things trans sites, where fire has gone from spreading in cis, or near its site of location, to trans, out to other chromosomes. And so we wanted to take a look at these trans sites and ask if fire is really there. And there's 2 possible models that could be going on here. One could be that the RNA sneaks off the X chromosome and somehow tucks into these other trans sites and we just can't detect it. But the method of detecting these things is very sensitive and can actually detect 1 molecule. So that's probably not the case, but it still could be. The second case is that you could have these chromosomes are actually in proximity to that locus. So in 3 dimensions, the nucleus doesn't have just linear chromosomes stacked on each other. It's like a bowl of ramen noodles. And so we could imagine that maybe RNA helps stitch some of those multiple chromosomes together in order to have proper architecture of the genome in the nucleus. So to test that, we have lots of data. I'll just give you the simple version of it. Is we can take, since we know fire is in proximity to each of those 5 sites, then we should be able to see those 2 sites near each other as well. So if we label 1 chromosome trans site in green and 1 in red, we get yellow, which indicates that indeed fire is actually near where those other chromosomes are in 3-dimensional proximity. What's a little bit bizarre about it is that there's 2 X chromosomes here, or 2 sites, but only one of them overlaps with fire. This happens in male cells because there's only 1 X chromosome, but it also happens in females even though there's 2 X chromosomes. So it could be, we don't know why that's happening, but it could be that maybe since the 1 X is from mom and from dad and 1's shut off, maybe this is a way of communicating who's been shut off. We don't know, to be honest, but it's an interesting theory that if this is either random, that the X chromosome picks mom or dad's allele at random, or it might be picking mom or dad's allele specifically. And we see that in biology quite often, there's dozens of examples where mom's genes are on and dad's genes are off and vice versa. It's called imprinting. And so we wanted to test fire in this case if it's important for this localization. So we made a knockout, which is, again, very laborious, but we deleted the region that contains fire, and you no longer get these localizations together. So in other words, in the presence of fire, these chromosomes are together. In the absence of it, they dissolve out. And it's actually very bad. These are stem cells, and they start to get very, very sick very quick. So we've just made the mouse of this and trying to see if it has an interesting phenotype. Ironically, we found this in adipocytes, it works in a stem cell to regulate genes that are involved in adipogenesis, even though it's not making a fat cell yet. So we don't know if this is maybe a pre-set up little structure to help the cell regulate adipogenesis at some point. And it's also highly expressed in the brain. So I think I want to introduce you to a sort of a new idea that you can have, that the nucleus is in 3 dimensions, and you can have RNA as nuclear beacons that can bring things together and stitch together multiple chromosomes to provide the right shape. Because the genome, the same genome is present in every single cell in your body. It's really the shape it takes on inside the nucleus that defines whether it's going to be a liver cell, a brain cell, or a kidney cell. And more importantly, if that shape is broken, that's when disease happens. So if we can figure out how to make those shapes, we can think of ways to reverse engineer this process and fix the genome and put it back together. I like to think of it as the little, it's like genomic origami. The stem cell can make anything like a paper airplane, or it can make one of these cranes. And in disease, you might have a crane with a broken arm, but if we can figure out how the genome's organized, we can then bring that arm back up and save those cells. So the good news is there's lots more of these. This is Arjun, my good friend and collaborator at University of Pennsylvania, and there's more fires. This is all new data that nobody else has seen but you guys, and I like to think of these guys as polka dot cells, where there's lots of these things where there's more dots than there are chromosomes. And there are these all the protein business happens in the cytoplasm. That's where proteins are built. These guys never leave the nucleus. In fact, they rarely leave their site of where they're made. So they might be serving as little lighthouses to help organize this massive job of packaging DNA into the nucleus. So this is what I'll wrap up with, is this idea that the nucleus is not random. It looks random. It looks like a bowl of spaghetti, but there's compartments within it. We talked about, this compartment is probably the most obvious one. It's called the nucleolus. It's the region of the nucleus that doesn't have any DNA in it. But what it does have is the ribosomal RNA, that thing that makes the protein, the RNA gene that makes the proteins, is those RNAs are made in a specific part of the nucleus called the nucleolus. And multiple chromosomes come together that contain these ribosomal RNA genes, and they're all made and dumped into this sort of vacuole here called the nucleolus. Splicing, similar. When things are starting to splice, they come into a certain proximity to a nuclear structure, again, driven by the presence of an RNA. The X chromosome that we mentioned was pulled off to the periphery is done so by grabbing hold of the RNA. It grabs the RNA that's caking that X chromosome, and it gets pulled off to the nuclear envelope or to the outer ridge. Again, RNA indicating position. And what we saw today is that we've known that RNAs can bend DNA within a chromosome, but it can also shape interactions across chromosomes as well. And all 3 of these things have one thing in common. And I think it's sort of the unifying, this is my new favorite way of thinking about this, is that there's sort of a unifying principle here of proximity. RNA indicates a location. It indicates proximity. In all of these processes here, RNA is simply bringing things into a specific location so that in helping to organize it. So I think RNA might be a really powerful player in the way we organize our nucleus and the way DNA gets packaged into the nucleus, and can do so just by being at its site of where it's supposed to, or where it's caking regions where it is as an indication of I'm here, I'm active, somebody come stick to me. Another way to simply think about this is a way of RNA traffic control, that airplanes actually don't know where they're going, right? They're sitting there flying around in the sky. It's really the air traffic control tower that helps keep this seemingly disorganized mess organized, where all these airplanes are trying to get to the right place, the right time, and there's thousands of them. It's kind of scary thinking there's that many in the air at once. Luckily, the clouds are there, so you can't see around, but but the airplanes don't really know where they're going, and it's amazing that they don't crash into each other, but it's really this air traffic control tower that's saying, hey, you go here, reroute here, circle around the airport for 2 hours, do whatever. And RNA might be doing something similar in the nucleus. It might be sending air traffic control signals that can be quickly turned on, quickly turned off to reroute and organize what is seemingly a similar chaotic mess within the nucleus. So I didn't have time to tell you everything about RNA, but it does a lot more. It's impressive that it's essentially involved in every process we've ever investigated in, and can play a wide range of roles. And I think this new approach of using the combination of genetics and mapping is really going to lead us to the gold quick, and hopefully improve human health and care in the near future. So to summarize, there's lots of long non-coding RNAs. How many are functional is not my business. I just want to find the ones that are. Human health and disease, I hope to show you that you can use the human genetics, find the equivalents in mouse, and start to learn which ones will be important for which diseases. And this new idea of proximity within the nucleus, that long non-coding RNAs might serve as nuclear organization factors. So in fact, the RNA in the junk drawer might be organizing the junk drawer itself. And with that, there's many people to thank. These projects are always done with tons of people and lots of collaborations, because the RNA leads us into all these mysterious new diseases and things, and it's really nice to have collaborators that are willing to work with us and figure out how these things are working. And really, it comes down to, I used to come to work every day to find an RNA gene, but now I come to work to hang out with these guys. They're really a talented group of people that really dedicate all their time and energy and effort, many hours, to finding these things, and have done a great job. So with that, I'll conclude and thank you guys.
All right, thank you, Dr. Rinn. Do we have